All right, so in this section we're going to talk about digestion. And if y'all remember, digestion is the process of breaking apart, macromolecules into smaller pieces. And so we talked about how there's mechanical and chemical digestion. And all along the digestive tract, there's some mechanical digestion that's occurring. So we said that mechanical digestion begins in the mouth with chewing or mastication, as well as the mixing of that food with your tongue, right? And then even when you swallow that bolus of food and it starts going down your esophagus and reaches your stomach, you still have some mechanical digestion in the stomach because the stomach starts churning, right? Once you enter that gastric phase, it starts churning and mixing that bolus of food around. Now, in this section, we're going to talk about chemical digestion, which we kind of talked about a little bit because we've said, yeah, you get some enzymes that are secreted by saliva. There's enzymes that are secreted by your stomach wall, so they're in the gastric juice. There's some enzymes that are secreted by your pancreas. But what we'll really focus on in this section are what are those enzymes doing, where are they secreted, and how are those nutrients absorbed once those macromolecules are digested, right? So with digestion, you guys, we say that it's catabolic. So if you all remember, catabolism was a form of metabolism, but catabolism meant the breakdown of larger macromolecules into smaller pieces, right? So for instance, you'd want to have catabolism of protein because protein's a large polypeptide, and if you have a catabolic reaction, what you can do is actually break that protein into smaller individual, let's say like amino acids that are more absorbable because they're smaller, and you actually can transport those across the cell more readily. So once you get the monomer, which is the individual unit of that macromolecule, like if it's protein, we say it's amino acids. If it's sugar, it would be like glucose, right, or fructose. Those are individual monomers of the macromolecule, and those are small enough to be absorbed. And we'll talk about how this absorption works. A lot of them actually rely on transport mechanisms like facilitated diffusion carriers or even active transporters too. So enzymes we find, you guys, in these secretions along our digestive tract are the things that are involved with catabolism. So enzymes can catabolically break down macromolecules into those monomers. So for instance, let's say if you eat some, a peanut butter and jelly sandwich for lunch or something, okay? And there's a lot of sugar in that jelly. Some of that sugar could be in a sort of longer chain of sugars. And so what you'd want then are enzymes that can break that sugar into smaller pieces that you can actually get absorbed across your cell wall, or not cell wall, but plasma membrane rather. Okay. Now we talk about these reactions as being hydrolysis, where you're actually using water to break the chemical bonds between these molecules. Okay. So we're going to first talk about the digestion of sugars here, and we said that it begins even in the mouth because within the mouth you find enzymes that can break down sugar, and specifically we talked about salivary amylase. Now back in the day we used to do a lab here where like students would donate their spit, and they all spit into this big old spittoon, you might say, or like a big old beaker of saliva, and then we would test the effectiveness of that saliva in terms of its digestion of sugar. It was actually kind of cool because you actually can see how well saliva was able to break down the sugar. We don't do that one anymore. I'm not sure why. It was kind of nasty though because you're talking about like collecting like a big old beaker of spit. You know what I mean? So, but we talked about salivary amylase, so it's actually incorporated into saliva. Now amylase breaks down starches, right? So starch would be, a polysaccharide you'd find in food like potatoes, that kind of stuff, right? Now, they're polysaccharides, so they're sort of a long chain of sugars. And what amylase does is it breaks down that polysaccharide, right, through hydrolysis and catabolism into individual sugar molecules. So theoretically, if you left a piece of potato in your mouth over time, it should get sweeter over time, right? Because the enzymes in your saliva start breaking the longer chains of sugar, which you can't really taste, into smaller individual sugars, which actually taste sweet, right? And so one of the tests that I used to do at a kind of a science visit, we used to go to like high schools and, convince people to go into science kind of thing, is we have the kids put a little saltine cracker in their mouth and they just would hold it there, right? And if they're patient, they would hold it and they wouldn't swallow it. But over the course of several minutes, what you'd find is that saltine cracker actually gets sweet over time because the salivary amylase in your saliva actually breaks down the starch in that to individual sugar molecules that taste sweeter, which is kind of cool. But it's also kind of nasty because it gets mushy over time, and you just have like this wet mush that you're holding on your tongue for a couple minutes. It's just like, all right, Yeah, it's because it's kind of sweet now. All right, good. I get the point. So we also talk about pancreatic amylase, right? So there's amylase that's secreted by the pancreatic juices. And so that's also going to participate in some further digestion of those sugars. Now, you don't actually get the absorption of those sugars in significant quantities until you get to the small intestine, right? So even though you're breaking down sugar within your oral cavity and you're breaking down sugar in the duodenum, you don't absorb a lot of those sugars until like the distal part of the small intestine or the middle of the small intestine, like your ileum and jejunum, right? So what this slide is showing here is you can break down larger sugar chains into smaller, more absorbable pieces like lactose, maltose, and sucrose. And these can be actually further digested by other enzymes. Okay, so lactose is something that we'll cover in lab today. But that's actually the formation of two different monomers, galactose and glucose. And we have enzymes called lactase that can break down lactose into these two sugars, right? Now, if you lack the enzyme lactase, which 2/3 of the world's population does, by the way, then you can't break down lactose into galactose and glucose, which means you can't absorb that sugar because you can't absorb lactose. So for those individuals that don't make the enzyme lactase, and they can't therefore break down lactose into glucose and galactose, that sugar is sort of at the whim of their gut bacteria, right? Because their gut bacteria can break that down for them. And so what you find is that people with lactose intolerance, if they ingest lactose, well, they can't absorb it, but their bacteria can break it down. So they get a lot of like gas and flatulence and sort of irritation from the bacteria that can feed on that nutrient that they can't absorb, right? It's kind of weird to think that 2/3 of the world's population is lactose intolerant. So it's interesting. We'll talk about that later too. So maltose is just two glucose monomers in a chain. So maltase breaks down maltose into 2-glucose, and you can absorb glucose across the intestinal wall. And then sucrose is fructose and glucose together. And you guys may have heard of fructose before. There's like high fructose corn syrup, right? Fructose is also a sugar you find in significant quantities within fruit, hence the name fructose, right? Now, what's interesting is that fructose is processed a little differently than glucose, but they still yield ATP, through metabolism. And what we find though is that fructose actually makes you hungry, right? So what if you're ingesting a lot of food that's high in fructose? Well, that increases your appetite, so you end up overeating. This is the problem with like foods that have high fructose corn syrup, is that it actually ends up making you hungrier. So you end up eating more than you normally would if you had a food in the absence of fructose, right? That's why it's a problem if you're like adding high fructose corn syrup to your food. So, but this kind of makes sense though with respect to fruit, like biologically, because if you find fructose in fruit, and fruit's a seasonal thing, you wouldn't want to get full on just like one apple because there's a lot of apples available, right? And so it would make sense then that to have this sugar that's in a fruit that's seasonal and it's only going to ripen like once a year or so, right? You're going to want to gorge on that and eat a lot of it. So it kind of makes sense that fructose makes you hungry, right? But you want to avoid that as a food additive because it could make you overeat, rather. So then it's associated with things like obesity. Okay, cool. So in talking about protein digestion, you guys, we said that protein digestion begins in the stomach. So do y'all remember the enzyme that's released that's involved with protein digestion? What's that called? By the stomach wall? Yeah, pepsin, exactly. Good. So it's released in the form of pepsinogen, but then it's activated into pepsin. You guys remember what activates pepsinogen into pepsin? Hydrochloric acid and pepsin itself. Good, nice. So those two can activate pepsinogen into pepsin, and you see that's what's going on here in the stomach, you guys. So pepsin is it's a protease. It breaks down these proteins, and so it breaks down proteins into larger polypeptides. But that's not the end of the story. So once this acidic chyme enters the duodenum, we still get lots of proteases that are released by the pancreas. And if y'all remember, the pancreatic proteases are released as proenzymes. So they're not in their active form. So if y'all remember, we talked about like trypsinogen and chymotrypsinogen. And because they ended with ogen, they were inactive. And we already talked about why that's important. And you don't want the pancreas to release active enzymes because it can auto-digest itself, right? So what happens then is that these proenzymes that are released by your pancreas, they're activated by the brush border enzymes in your duodenum into like trypsinogen and trypsin and chymotrypsin. And now those enzymes, what they could do is they break down these larger polypeptides into smaller polypeptides, right? And so this will continue until you break these down into individual amino acids or dipeptides. That's what you're able to absorb. So you're able to absorb either two amino acids together, it's a dipeptide, right? Or individual amino acids, okay? Because they're small enough to actually transport into the cell. And you actually use transporters that are in the membrane of the cells that line your duodenum and small intestine, and they can pump those into the cytoplasms of those cells, where then they're further transported into the bloodstream, okay? So you have to break it down to absorb them. So you want to break them into individual amino acids. This begins in the stomach, continues in the duodenum, and then you absorb these in the small intestine. So it makes sense. So if we look at this process here, guys, here's the long polypeptide here. And we can have enzymes that can break this polypeptide into individual amino acids, which you can see here. And do you guys see that you co-absorb sodium with this amino acid? Now, if you'll remember that one's facilitated diffusion. And so for facilitated diffusion, you're basically allowing the diffusion of a molecule into a cell down its concentration gradient. Now, if you guys remember, sodium and amino acids can't normally just cross the plasma membrane because, for one, they have a big charge if you're sodium, and two, the molecule is too large if you're an amino acid, right? So what you need then is a transporter protein within the membrane of these cells that can allow these things to actually flow down their concentration gradient into the cell, okay? And then we also have amino acid carriers on the basolateral surface that allow for amino acids to get absorbed into your bloodstream. And all this occurs pretty quickly. So that's kind of interesting, though, is that you absorb sodium and amino acids simultaneously, right? And so what you'll find is there's a lot of like co-transport in digestion, and when we get to water, it'll help make more sense too. All right. So what about lipids? Well, lipid digestion actually begins in the mouth because we have salivary lipase, and so you can actually start to break down lipids even within the oral cavity. I think we talked about this already because we said how like if you eat like a fatty meal, and sometimes you can feel like the greasy food kind of coat your mouth still, and it continues to be digested there, so it kind of like tastes differently, almost like your mouth's like a little more sticky because it's so fatty. Maybe you guys haven't thought about it before, but apparently I've noticed. So you have lingual lipase, right? So it's actually breaking down lipids there. So lipids would be things like phospholipids, triglycerides, cholesterol, right? And these things are going to be broken apart by lipases. So lipases actually will break down triglycerides into individual fatty acid chains. Okay. So a triglyceride, if y'all remember from AMP1, was a glycerol backbone and 3 fatty acid chains stuck to that. And then lipase can basically break off that backbone, and then you can separate the fatty acid chains individually, which are more absorbable. Okay. Now we also have gastric lipase as well, and so this continues within the stomach. Now then we have bile, right? Now, do you find enzymes in bile? Do y'all remember? Are there enzymes in bile? No, exactly. So what's the purpose of bile? Do y'all remember? What's bile good for? So what do we use bile for? If there's no enzymes in bile, what can it do for us? Let me know you guys find the answer. I'm going to drink some water. Good. It breaks apart fats. Good. And the term we use to describe breaking apart fats is called emulsification, right? So emulsification. So to emulsify means to break apart fats into smaller individual pieces. But that doesn't involve enzymes. It actually involves detergents, which are the emulsifiers like the bile salts, okay? And so what happens then is to emulsify effectively separates fat globules into smaller pieces. And I mentioned to you guys how, let's say, you guys have all seen this, by the way, because soap is also an emulsifier. And so you guys all use an emulsifier this morning to wash the oils off of your skin and out of your hair, okay? And the way this works, or let's say if you washed a pan or something from dinner, you know. So let's say if you pour water into a pan, and if, let's say if there's a little oil in that pan, if y'all notice, have you guys ever noticed where there's like oil that floats on the surface of the pan? And it might be like kind of like in a big glob or something initially, right? But then what happens when you pour soap in there? What happens to all that oil? You all ever seen that before? It just separates, it goes, and it separates fast. And if you ever really looked at it when it happens, you'll notice that when it separates into lots of really tiny little balls, right? And it spreads off the sides. And so that's what soap does as an emulsifier, is it breaks apart that piece of fat or oil into lots of smaller pieces. That's effectively what bile does within your body to the food that you ingested, right? But only to lipids. So bile's an emulsifier of lipids. So it breaks apart those lipids into smaller pieces. But what that does, though, is it actually increases the surface area for digestion. So you're not physically breaking like chemical bonds, like covalent bonds on these fat molecules. You're just separating the fat group of molecules into smaller pieces, like divide and conquer, right? Like breaking apart these pieces into like smaller groups, and which are more readily digestible, okay? And so this digestion then also continues by the lipases that are released by your pancreas. So these pancreatic lipases can help digest these lipids even further, right? And so once we get things like monoglycerides or diglycerides, those are more readily absorbable across the plasma membranes of your digestive tract. Now, I'll ask you guys this from AMP1. Can lipids readily cross the plasma membrane of your cells? Do you remember simple diffusion? What was simple diffusion across plasma membranes? You're good. It's diffusion. So it goes down its concentration gradient, right? So you're going from high to low. So it might be high inside of the digestive tract and low inside of the intestinal cell, right? So then it flows across the membrane. But if it's called simple diffusion, did it use protein transporters to cross the membrane? No. It just flows right through, right? And so what we find then is that lipids don't need transporters. They just get absorbed by your cells, right? Because they're lipids, they could just flow right across the plasma membrane. If y'all remember, that was simple diffusion. So if we look at this process here, you guys, here's our larger fat globule, right? And then here's our bile salts. Now, what's kind of fascinating about these bile salts is that they're amphipathic. To be amphipathic means you have a polar end and a nonpolar end, okay? So what happens is that the nonpolar ends of the molecule associate around each of these little fat globules. Then the polar ends of the molecules associate with water. But effectively what that does is you can break apart this fat globule into lots of smaller pieces. Now, if you guys have taken like, let's say like geometry before, you know, and you start doing things like measuring like the volume of something or the surface area of something, right? Or even like pre-calculus, you do that too. So what you'd find is that there is actually more surface area in the sum of all of these individual pieces than there is in this larger fat globule. So even though it looks like a large fat globule, if you calculated the total surface area on that one fat globule, it would actually be less than the sum of the surface areas of all these pieces. So what bile does is it breaks down the fat globule into smaller pieces, but increases the surface area. And what that does then is it makes more area of these lipids available for the digestive enzymes to come in and start chewing it up, right? So that it's easier for lipases to get in here and start basically breaking up these lipids into, you know, more absorbable pieces. That's the whole purpose of emulsification. So what if you didn't have bile? What would happen to you guys?
Digest fat as well?
Right, definitely wouldn't digest fat as well. And so let's think about why that would be though. So if you don't have bile, because maybe there's a bile duct obstruction or if you have liver failure, we know you wouldn't digest fat as well. And we know that bile salts emulsify. Could you still digest fat though? Yes, good. So what would be the difference between having bile and not having bile in terms of digestive speed? What's that? Not as fast. Exactly, you got it. And it's not as fast because, why is that? Nice, good. There's less available surface area in larger fat globules. So you can still break down the fats, but it's going to be slower because those enzymes just have less area to access. Very good. Awesome, you guys. So what we find here, you guys, is that these lipids, then, once they're broken down into smaller things like monoglycerides and diglycerides, they actually form something called a micelle. And so a micelle is almost like a vesicle. So if you guys remember like a vesicle inside of a cell, it was a double layer of phospholipids, and there was an aqueous center. A micelle is actually a single layer of lipids, and their fatty acid tails all protrude inward. So what you find is that they're like the phospholipids that form a little sphere, but their fatty acid tails protrude inward, and the center of that little sphere is not water-filled. It's filled with lipids, right? That's what a micelle is. In fact, pharmacists can use micelles to deliver drugs across your skin because micelles are absorbable across the plasma membranes of your cells. So let's say if you had a fat-soluble drug, like any of the fat-soluble vitamin-derived drugs, like the retinoids, right, which are derived from vitamin A, those could be packed into micelles and then delivered in a cream on your skin. And so because you can absorb micelles across the plasma membranes of your cells, that means you can actually get that drug to cross your skin barrier and get into your body where it can go into your bloodstream, which is kind of cool. Now in your digestive tract, though, these micelles basically just absorb across your membranes into the inside of these cells. Now what's pretty cool, though, is that the inside of these cells further process those micelles into lipoproteins, right, which we call chylomicrons. So do we have that? Do we go into that level of detail? I guess we don't go into that level of detail. Never mind. So you guys just need to know at this level here, where micelles are absorbed across the plasma membrane, and then they're further processed by these cells into chylomicrons. Now, are lipids transported by your bloodstream or by your lymphatic system when they're absorbed in your gut? Lymphatic, you got it, right. And what's the mixture of lymph and fat called? Kyle, right? C-H-Y-L-E, right? My name, Kyle. But C-H-Y-L-E, that's how it's spelled, Kyle. So what you're seeing then, you guys, is that those chylomicrons, when they're absorbed in the lymph, they form fatty lymph, which is called chyle, right? It's spelled like chile, okay? But effectively, this lymph then goes back towards the heart, and then it mixes in with your blood. So what's interesting then is that lipids are not processed by the liver first. Like every other nutrient that we talked about that gets absorbed by your blood, that goes straight to your liver because of the portal system, right? Now, lipids bypass the liver. They're not part of the portal system, and they actually dump in by your heart where the lymphatic system drains. And that's an important note because we're saying that when you first absorb lipids, one of the first organs they encounter is your heart. And that can kind of associate with things like cardiovascular disease. Okay, so what about nucleic acids? Well, we have basically like ribonucleases and deoxyribonucleases, and so we just call them nucleases. And these enzymes break down DNA and RNA, right? So what's kind of cool then is if you're eating, plant material or animal material, not only are you breaking down the plasma membranes of those cells and getting some of the protein and sugars from the cytoplasm, but you're also breaking down the nucleus of those cells, breaking apart their DNA and RNA, absorbing that DNA and RNA, and then you're actually using that for your own genetic material. So you literally are what you eat, right? Like if you take anything out of this lecture, like you are literally what you have eaten in the past, which is kind of weird to think about. So I like to think about, well, what am I made of if I am what I eat? Well, I used to eat a lot of Wendy's chicken sandwiches, so I'm a lot of Wendy's chicken sandwiches, you guys, that's for sure. But I'd say in the last 10 years, probably a lot of beans and a lot of chili. A lot of chili, right? So good times. So so you're gonna break down these nucleic acids into smaller, nucleotides, which you can absorb. They go into your bloodstream, and you can use those for your DNA. You can use those to make ATP, so on and so forth. Okay. So where does most absorption occur, you guys? We talked about this. Small intestine, nice. So most absorption occurs in the small intestine. And what was it about the small intestine that made it so absorptive? Why is it so special? You guys remember? There were specialized features of the small intestine that made it an absorptive organ. Nice, good. The microvilli and the villi and the intestinal folds. So there's like ridges on top of ridges on top of ridges. And all of that, again, increases the surface area inside your small intestine. And because there's more absorptive surface, you're able to really get those things into your body, right? Now, it's not to say that there's no absorption in the stomach or the mouth. It's just that it mostly occurs in the small intestine for two reasons. For one, there's a lot of absorptive surface area. And two, that's also where these nutrients are going to be mostly broken down, which means that they're then more readily absorbable, right? So you can still have some like lingual absorption. You still absorb things from your mouth. You guys heard of like sublingual administration of drugs, So it's not to say that the mouth can absorb or the stomach can't absorb, but most of it occurs in the small intestine. So we're saying, all food and 80% of your electrolytes in the small intestine. Now, most of your water is absorbed here as well, you guys. And what's really cool too is you can recycle these bile salts. So do y'all remember how much bile you can make and secrete in any given day? Yeah, about the volume of a water bottle that I have here, which is almost a liter. So I think our book said about 900 milliliters or so, which is 9/10 of this water bottle. Now, within that, bile is going to be a lot of water, but you also have bile salts. And your body used energy to make those bile salts. And so what's really cool, though, is that you can reabsorb those back into your bloodstream, and then effectively your liver can absorb those from your blood and then recycle those back into bile. So, there's not a whole lot of waste here, you guys. You're actually going to reuse these things to digest fats even further, right? Now, most of nutrients that you absorb, you guys, are absorbed by active transport. So if it's active transport, does that require ATP or does not require ATP? It does require ATP. Good. So what does that mean about absorbing nutrients? It means you got to put a little bit of energy in to get the energy into your body, right? So it's almost like you gotta like, I don't know. You might say that you just gotta like use a little bit of energy to get nutrients into your body, but you obviously get more energy out of those nutrients because if it took more energy to absorb them than what you'd get, you'd have a negative energy balance, and obviously you couldn't survive that way. So it makes a little bit of sense. Now, water though is not absorbed by active transport because there's no such thing as a water pump in your body. So you can't like use ATP to pump water. That doesn't exist. Water only moves via osmosis. And if you'll remember, osmosis is the process where water goes from high water concentration to low water concentration, or you can say that it follows solutes, right? So as you're absorbing sodium and glucose and amino acids and nucleic acids, and you're absorbing those solutes into the cells, because you're altering the solute concentration across the membrane, water will move in the direction of solute absorption. So as long as you're absorbing solutes, you're going to absorb water with that. That's important. Keep that in mind, you guys, because you'll come back to that in pathophysiology as well. Now, one of the other nutrients that you don't use active transport to absorb, though, is lipids. Because remember, lipids was simple diffusion. They just absorb across the membranes of your cells, no problem, right? You don't use ATP. They just they just go down their concentration gradient and cross the membranes of your cells, if y'all remember that process. So in terms of vitamins, you guys, we get a lot of absorption of vitamins in our small intestine, like the fat-soluble vitamins like A, D, E, and K are carried in micelles. Because remember, these are fat-soluble. So you find these fat-soluble vitamins. I like to think of this as CADE, like K, A, D, and E. For some reason, my brain remembers that well. So CADE are the fat-soluble vitamins, or you could say DEAK, D-E-A-K, right? Whatever we want. You can mix it around any way you want. But because they're fat-soluble, or lipid-soluble rather, you find them in the middle of a micelle. So when you absorb a micelle across your membrane, those fat-soluble vitamins come with it, okay? And so then they can just diffuse then in your bloodstream from there. Water-soluble vitamins are absorbed by diffusion or passive or active transporters like vitamin C and B. And then B12, you guys remember this one? What do we need to absorb vitamin B12? And it's made by your stomach. Something that's made by your stomach is absolutely essential for the absorption of vitamin B12. Do you remember what the thing was? So if you don't have this, you can't absorb vitamin B12. And the example we gave was conditions like alcoholism, where because alcohol is irritating your gut, you don't make this molecule, which means you don't absorb vitamin B12. But vitamin B12 is necessary for nerve function, so you can get dementia from the deficiency of this molecule. It's a glycoprotein, and so this specific type of glycoprotein is called, starts with an I. Intrinsic factor, yeah, exactly. Is it up there, you guys? Is it on the slide?
No.
Okay. Actually, yeah, it is. Intrinsic factor. Good morning. So, vitamin B12. It's absorbed by intrinsic factor. So, intrinsic factor is this glycoprotein made by your stomach. Vitamin B12 binds with it, and then you absorb both together. So, you got to have this complex to absorb vitamin B12. If it's necessary for proper nerve function, how important do you think that is for your body? Very important. Very important. So what I haven't figured out, I'm really curious to know about, is what do people do if they've had their stomachs removed? You ever those situations? Like there's a case maybe 15 years ago where there's someone who tried to kill themselves by drinking bleach, and they basically burned their esophagus and stomach so severely that they actually had to remove the esophagus and stomach, and they took the small intestine and brought that all the way up to the pharynx and reattached it. So they didn't have an esophagus or a stomach anymore, and there was just a small intestine going up to their pharynx. So my question then in those cases is, what do they do about vitamin B12? Now, my guess would be that they might be getting vitamin B12 injections, like subcutaneously or something, sort of, on a regular basis. Or they might be getting like, exogenous or outside their body intrinsic factor. Like you can take pills of intrinsic factor with your food, which can help you absorb vitamin B12. That would make sense. Otherwise, they wouldn't be able to make it, which means if you didn't have a stomach, eventually your nervous system's going to break down. And you get things like Korsakoff's dementia. So it's kind of fascinating. All right, guys, cool. In the large intestine, you guys remember we talked about how your gut bacteria can make vitamins like vitamin K and B complex. So vitamin K, it's made by your gut bacteria in your colon, which means it's also absorbed in the colon. So you absorb vitamin K along your large intestine, or colon. You also absorb B complex vitamins from your colon as well. Now, vitamin K is necessary for blood clots. B complex vitamins are necessary for metabolism, right? So that's also extremely important. We're saying that you need a large intestine to absorb sufficient quantities of vitamin K to form blood clots, and you need a large intestine with good bacteria to make B complex vitamins that you can absorb to basically keep your electron transport chains working. So a lot of these B complex vitamins, you guys, they're actually the cofactors in the electron transport chain for oxidative phosphorylation in your mitochondria, right? So you need B vitamins for energy. If you didn't, then you can't make enough ATP to live. So that's pretty essential. And so there's some ideas here behind gut flora and energy levels, right? Like if you're feeling very lethargic, maybe your gut flora aren't happy and they're not making enough B vitamins for you. So it's kind of fascinating. What they're finding, guys, is a lot of connections between the bacteria in your gut and the health of the rest of your body. And there's some really interesting things that I'd like to get into with you guys right now, but we would talk more about in a class like pathophysiology, where it's more relevant to disease. Okay, so for electrolytes, you guys, ions are actively absorbed along the length of your small intestine. Iron and calcium absorbed along your duodenum. Sodium is coupled with glucose transport as well as amino acids. Chlorides transported actively. We have potassium diffusion down osmotic gradients. And usually what you absorb in your small intestine is what you're going to absorb for the rest of your body, if that makes sense. So one thing that's important to note there is about water is that water is only absorbed by osmosis. So what if you had an absorptive disease like Crohn's disease or ulcerative colitis, where you have a lot of inflammation within the small or large intestine, and that inflammation reduces the surface area, and you don't absorb these nutrients as well, right? What's going to happen to your feces? Are they going to be solid or are they going to be liquid if you're not absorbing nutrients across the digestive wall? Liquid, right? So we call that osmotic diarrhea. And the reason being, you guys, is that if you're not absorbing those nutrients, you're also not absorbing water. And so if someone has nutrient absorption disruption, right? If they have dysfunction of that, rather, then they're going to have watery stool as well. That comes with that. So they're basically pooping out like nutrient-rich feces, and the water goes with it because you need to absorb those nutrients to get the water to go with it because it's osmosis. Okay, cool. So iron and calcium, you guys, are absorbed along the mucosal cells with something called ferritin. Now, when you need them by your body, there's a molecule called transferrin that helps to transport calcium and iron around your bloodstream. And you might wonder, well, why do you need a special protein for calcium and iron absorption? Well, free iron is actually toxic to your bodies. So if iron is not bound to other molecules, if it's by itself, it ends up helping to catalyze the production of what are called free radicals. And free radicals can just break bonds indiscriminately all around your body. So free iron you don't want in your body. And that would make sense, how you need protein to bind to iron. That way it's not free, because if iron's loose, it starts causing problems inside your body. Okay? That's why you actually can have iron overdose, especially with conditions like hereditary hemochromatosis, where they don't transport iron properly, and it leads to excessive free radical production. And again, we would come back to that in pathophysiology. Now, for calcium absorption, it doesn't lead to the production of free radicals, but calcium is really important for things like blood clots and proper nerve function and secretion. And because calcium levels are fairly low inside of your body's cells, you wouldn't want to alter that too much. So by absorbing that calcium and having protein grab onto it, you can maintain normal blood calcium levels and intracellular calcium levels. Because you don't want to like overload your body with too much calcium too quickly, because that can cause a lot of problems. Okay, so we also know that calcium absorption is regulated by parathyroid hormone. We talked about this in the endocrine system, right? So we said that when your blood calcium levels were too low, the parathyroid glands behind your thyroid gland, they have humoral sensory input, and they can basically release parathyroid hormone. And so what parathyroid hormone does is it acts to increase blood calcium. Do you guys remember the 3 ways that parathyroid hormone does that, by the way?
2 with the kidneys.
So it helps with calcium reabsorption in your kidneys. Good.
The osteoclasts.
It helps to stimulate the osteoclasts of your bone to break down the bony matrix. Good. And then the third is with the kidney. So one was calcium absorption in the kidney. Another one was bone breakdown. And the third one, kind of counterintuitive, you guys, it stimulates the production of vitamin D by your kidneys. Yeah. So remember, your kidneys can make vitamin D3, which is effectively a hormone. But what parathyroid hormone does is it stimulates your kidneys to make that hormone. And so what vitamin D does, though, is it allows for calcium absorption by your gut, because vitamin D stimulates the production of these transport proteins like ferritin and transferrin. So it makes sense. Cool. Good job, you guys. Okay. So for water, you guys, you absorb about 9 liters of water across your digestive tract a day. That's a lot of water. Think about it, right? That's 9 of my water bottles here. This is a 1 liter water bottle. 9 liters across your digestive wall. So you might wonder, where does that water come from? Well, you probably aren't drinking 9 liters of water a day. You might. I mean, that'll be a tremendous amount of water, right? But it's mostly in the food you eat, because the food you eat is not dehydrated. You know, there's a lot of water in that food. You're also getting water in other ways too. Like for me, I get a lot of it in coffee. Like I probably already had a liter of French press coffee this morning. You know what I mean? Like so. But so water absorption is through osmosis, right? So it's only going to follow the solutes. Now, most of it's absorbed in your small intestine. The rest of it, you guys, is absorbed in your large intestine. So that's where the dehydration of the feces comes into play there in your large intestine. So net osmosis only occurs if there's a concentration gradient, and you only get that concentration gradient if you're actually absorbing nutrients. Okay? So water uptake is coupled with solute uptake, and that makes sense. So if you're not absorbing solutes, you're also not absorbing water, and that leads to diarrhea, right? So it makes a lot of sense there.